A lot of us probably less introduction. Where did I stay?
Good afternoon. I'd very much like to welcome you to this year's ASHG Pacific Biosciences Workshop. My name is Jonas Korlach. I'm Chief Scientific Officer at PacBio. Thank you all for coming to our workshop today. I'd like to begin by expressing our sadness about the passing of Dr. Alan Roses about 3 weeks ago. Dr. Roses was planning to speak at this workshop, and as we mourn his passing, we would like to commemorate Dr. Roses. Alan Roses was a true giant in the field of human genetics, with so many fundamental contributions and discoveries that are too numerous to list. Alan Roses was one of the first clinical neurologists to apply molecular genetic strategies to neurological diseases. His laboratory reported the chromosomal location for more than 15 diseases, including several muscular dystrophies and Lou Gehrig's disease. He led the team that in 1992 identified ApoE as a major, widely confirmed susceptibility gene in common late-onset Alzheimer's disease. I also want to mention his founding of and leadership role in the SNP consortium that initially published a catalog of single nucleotide polymorphisms that were later used to map susceptibility loci during the GWAS era. We would like to dedicate this workshop in his memory as we remember his life's work and achievements. We had scheduled an interview with Dr. Roses at his office at the end of September, which turned out to be just a few days before his passing. I would like to ask you to join me in remembering Dr. Roses in a moment of silence and the following tribute to him, as he influenced not only the field of human genetics, but also hundreds of researchers, of students, and of postdocs as a mentor, as a collaborator, and as a friend.
My name is Alan Roses. I've been at Duke University Medical Center for 46 years. And during that time, became a genetics researcher in 1979 when reverse transcriptase was published, and I knew there was a new way of getting at the genetic diseases. So in the course of going from linkage, discovering ApoE, which was highly significantly associated with Alzheimer's disease, we're going down a path in which it really became necessary to focus more and more on things that might lead you to the mechanism of disease. I'm saying this history basically because it really is the important part of why we're currently now studying short tandem repeats. And so when we started applying this methodology to the search of SSRs in major diseases, particularly major neurological diseases, for which I can lay my hands on collections through people that I've met or trained over the last 40 plus years. What we found is sitting in the middle of the sequence were a whole bunch of poly-Ts. And again, make a long story short, that discovery became the TOM40 523 polymorphism, which is 100% informative in the population and allows us to map out by human phylogenetic mapping that these changes in the length of the poly-Ts change the age of onset distributions, but also related to different symptoms and different severities of disease. And we're more likely to be involved with the mechanism of Alzheimer's disease than ApoE. ApoE changed the rate of the development of the disease. And it became very obvious that they were interacting with each other. So now we had ApoE, fragments of which we knew bound to TOM40, other things bound to TOM40. Virtually everything binds to TOM40 because TOM40 is an open pore through which all peptides and all proteins that need to be made into mitochondria by fission have to come through. And so there's a all these different binding sites and things like that. And it led us to see if we could find the metabolic basis of Alzheimer's disease, and basically we felt we had. We found the first linkage to ALS. That turned out to be a SOD1. Our latest work that we're really excited about, because I think it's the worst disease in neurology, motor neuron disease, or ALS. We have findings that relate to a new SSV, or single sequence variant, in a region of one of the genes that is thought to carry 150 mutations. Long story short, what we found was that in the less than 200 patients that were being looked at, with each having an allele, so that's 400 or so alleles and affecteds, and another 400 or 500 controls. We found that the statistical significance was extremely high for the 17, 18 variants, those two at the other end. And 18, if we looked at it, the only homozygotes that we have found were in patients with ALS. And all the other patients that we found had 17 and 18, while in the controls, 16 and 15, and 17 ranked third in the weight of their poly-T lengths. It gets us closer to the mechanism. It gets us closer to alternative splicing. It gets us closer to be able to see what's going on in the disease, and that's where we are. When I went to speak to this dean down under, and I go to Australia a lot, but when I went to see this dean down under, I said, well, you know how my career has been. I find something, and it's 5 years before anybody does the next experiment. He says, yeah, we heard you call it the Rose's effect. Instead of patenting, you just publish it, nobody does it. But the reason is, first, they don't believe. Then they say, oh, that might be interesting, we're in it, and they write reviews about it. And then they, in some cases, they say they found it. But luckily, most of those cases where they found it have been all wrong. So.
I would now like to begin with the workshop presentations. We have a terrific lineup of speakers. Today, before I ask you and Ashley to take the podium, I'd like to spend a little bit of time, say a few words about what we've seen for smart sequencing, specifically in the area of human genetics. And overall, we've seen a real rapid increase in the number of peer-reviewed publications in all areas, not just human genetics. There's now over 1,800 publications that describe the aspects of smart sequencing in various different fields. Many of these are in the human genetic field. We're well on track to have over 1,000 publications this year, to a total of over 2,000. Currently, the rate of publications is about 25 to 30 new papers per week, which is on average about 1 new paper every 5 hours. And with regard to the human genome research, I want to specifically in this workshop highlight one aspect of genetic variation, that is structural genetic variation. Larger than SNPs, and you have various different forms, of course, deletions, insertions, tandem duplications, interspersed duplications, inversions, translocations, which we have known for a long time that they are very important to understand diseases, understand traits, understand the mechanisms of evolution, but of course they've been the orphan child in the human genetics field because of the technological limitations of the short-read technologies, which led the research community to largely focus on SNPs. We've known for 10 years since Craig Venter's genome that in terms of the number of bases, structure variation actually comprises the vast majority of genetic variation. 3/4 of all the bases that make you different from me are not in SNPs, they're in these structure variants. And so one of the phenomena that we've seen is that PacBio has been used in a variety of projects. Some of them have already published, and many of them are underway to generate high-quality de novo human genomes, to really understand the complexity of the human genome with regard to the variation that exists in terms of different ethnicities, and to start assembling a catalog of structure variants that are pervasive in the global human population. And so some of these papers have published. This is an example on Nature Communications of the first de novo assembly of a Chinese genome, identifying thousands of deletions and insertions, many of which are new. The graph here on the bottom shows for both insertions and deletions the power of PacBio sequencing with regard to structure variation detection. You can see that by sequencing just one individual with PacBio, you discover about a third of all the structure variants that had been identified with the 1,000 Genomes Project, sequencing 2,500 individuals with the Illumina platform. So there's about between a 500 to 1,000-fold higher sensitivity with regard to structure variants with PacBio. Very recently in Nature was a paper on the PacBio de novo Korean human genome assembly. This is the most contiguous diploid human genome assembly to date that was assembled and led by Macrogen, Professor So at Macrogen, with a contig N50 from the assembly of 18 megabases. So this genome is in pieces, half of the genome is in pieces that are gapless, contiguous stretches of sequence of 18 megabases or larger. And on the right here, you can see that, again, in this paper, numerous structure variants were detected, many of them new. And on the bottom, you see a figure that some of these structure variants are specific to the Asian population, several insertions that are not pervasive in other ethnicities. So then the question is, how do we go from these high-quality, reference quality genome assemblies to going towards the population? And on the right here is a table highlighting the number of structure variants that were identified from the high-coverage PacBio genome assemblies, and as a rule of thumb, there's about 10,000 deletions and 10,000 insertions identified in these individuals. And so on the left, you can say, well, I just have to sequence a lot more individuals, and now I'm going to get to the landscape of common structure variation across the global human population at the different variant frequencies of, let's say, 5, 1, or 0.5%. But we can take a play out of the playbook from the 1,000 Genomes Project, which, as you remembered, was sequencing a much larger number of individuals at fairly low coverage, only about 6 to 7-fold coverage. And this same thing holds true for structure variants, that if you do this, and instead of sequencing 100 humans at 50-fold coverage, sequence 1,000 humans at 5-fold coverage, that's the same total amount of sequencing, the same total amount of cost. You're doing much better in identifying the structure variants at these different variant frequency levels. This, of course, can then also translate to identifying structure variants in the individual, and this was pioneered by researchers at Baylor, Jim Lupsky and Richard Gibbs, applying 10-fold PacBio coverage, so very moderate PacBio coverage, in addition to the Illumina sequencing they had done, this is Jim Lupsky's genome actually, and demonstrated that that more than tripled the number of structure variants that could be found in that particular genome. So in the context of the new SQL platform, we decided to test this particular experimental design strategy on the SQL system. The sample we chose was NA12878 for the obvious reasons that it's very well studied. So you have extensive truth sets and validation calls that you can apply to see how well you are doing. There is a deletion and insertion call set from the 1,000 Genomes Project of Illumina. There's a deletion call set from the Genomes in the Bottle Consortium. And then there's a call set from Ali Bashir at Mount Sinai from a high coverage PacBio assembly. So we subjected the sample to PacBio sequencing on the SQL system, about 25 kb in average insert fragment size selected with the blue pippin from Sage to about 15 kb. Using the version 1.2 chemistry, we required 3.4 million reads with an average of 11.8 kb, that's the read length, and 50. So half of the reads were 11.8 kb or longer, and you see one example read length distribution here. And then the reads were mapped to the HG19 reference genome, and the variants were called with the PB-Honey spots software that was developed by Adam English at Baylor College of Medicine. So this is what we found, and compare it to the truth set. The truth set is the union of the 1,000 genomes deletion calls and the genome in the bottle deletion calls, and then also for Illumina the insertion calls. So you can see that with just 10-fold coverage on the sequel system, more than 7,000 deletions and more than 7,000 insertions were called. We're capturing about 80% of the truth set. And you can also see that, in addition, a large number of structure variants are identified that were not in the truth set. And more than 2/3 of those are immediately validated from the high-depth coverage PacBio assembly that was done at Mount Sinai. Here's the, on the right is the length distribution of those structure variants, which is in very good concordance to what we know about the overall length distribution in a human genome of these structure variants. And the only other point that I want to make about this is that you can see that most of the structure variants are in a size range from 25 or 50 base pairs to maybe about 7 or maybe 10 kilobases. So that puts a little bit less pressure on the quality of the library and the size of the library preparation. You could probably capture all of these fairly easily with, let's say, a 10 kb library, and it doesn't require the really ultra-long insert libraries that you would strive for in the context of de novo assemblies. So we had a blog post this morning talking about this structure variation call set, and it's been made available through a link on the DNA Nexus website. It's a featured project, and I would like to encourage you to go there. You can explore and look at those data yourself. Now, I do want to spend another minute in talking a little bit about tools. When you have a new technology, it takes a little bit of time to develop these types of tools. There's numerous structure variant callers now available for PacBio data. I already talked about PB-Honey. Parliament is another one from Baylor College of Medicine. Sniffles has been developed in the lab of Mike Schatz, which then makes it very straightforward to call these structure variants. One aspect that's shown on the left here is an aspect of visualization, and I want to highlight that. Of course, an ecosystem has developed around the short read platforms, mainly Illumina, with reads and alignments and variant callers, and the tools that have been developed were tailored for the short read platforms, and they may not immediately be optimal for the long reads. So I want to talk about one project that my colleague Aaron Wanger has spearheaded to adapt the popular platform IGV from the Broad Institute to long PacBio reads. So I'm sure you're all familiar with the IGV browser, and so here's a stretch of 1,000 bases, and you see the Illumina made pair reads, the coverage track, and then you see the variants here. Now, if you zoom out to a window that's relevant for PacBio reads, this is 10 kb, then the IGV viewer is not, in its baseline view, it's not that useful, because most of what you see is the random insertion deletions. The substitutions in the reads actually go away for both Illumina and PacBio data, and it's not all that useful. And so through simple filters and visualization enhancements, Aaron has worked with Jim Robinson, the author of IGV, to transform this view into this kind of a view, which is much more informative. So what was done here is to properly space the reads, to remove the random insertions deletions, to bring back at this zoom level the substitution errors, and to filter out the random substitution errors and only keep the ones that are in consensus, that are there at a particular position at about 20-25% that can be set by the user. And now you can see, and then I should mention that in addition, one other feature that was added to show large insertions and label them. So here, for example, is about a 400 base heterozygous insertion that you can now clearly see. And so you go from something like this, it's the same data, to something like that, which is much more useful for the researcher in actually seeing what's going on. You can immediately see that this heterozygous insertion is phased through the individual long reads and occurs only in the context of reads that also have this cluster of heterozygous SNPs here. And then one last feature, you can highlight one of these bases, one of these SNPs, and can sort the reads and immediately do what we call a quick phase by grouping by a base pair at a certain position, and now you can immediately see that these long reads are phased over long distances, and you can clearly identify the two haplotypes. So through these fairly simple adjustments, you go from something that's not tailored and not optimized for the particular read structures of PacBio reads to something that is very useful and that will immediately give you very nice visual impressions of structure variants. I'm just showing two examples here. This is a 2.4 kilobase homozygous deletion, where you can see those reads go through, and the deletion is clearly marked in the PacBio reads with the associated phased SNPs. Whereas in Illumina, if you have repetitive sequence in the reference, you often have these ambiguously mapped reads in the hollow bars that make the identification of that deletion much more difficult. Here's an example of a heterozygous insertion next to a SNP that was missed by the Illumina technology and very clearly now is highlighted in the IGV view of the PacBio data. So this IGV module is now available publicly through the IGV developers build. It will be fully released a little later this year. And we also have the demonstration at our booth, and Aaron is also there. He has office hours at 10 o'clock in the morning, and I think also in the afternoons. You can come by and get a feeling for that and can try it out yourself. So I want to spend a quick word about the current SQL performance and an update since we launched the system a year ago at ASHG at this conference. And over the past year, we have implemented a number of improvements on the library preparation, on the sequencing chemistry, on the analysis software. Initially, the availability of SMART cells, as you know, was quite limited, and so we've worked to remove that limitation, and we appreciate your patience as we've worked through these improvements. Having now reached a point with the latest chemistry where the system is very stable and consistently gives this kind of performance that you see here for all different library sizes. So on the left, you see a number of runs with regard to yield and read length of short insert libraries. These are amplicons, so you can see the overall yield per SMART cell. These are 6-hour runs, is ranging between, let's say, 4 and maybe 8 gigabases, and the read length N50 ranges from 17 to 20 kilobases, so half the reads are 17 to 20 kilobases or longer. And then also available now for long insert libraries in all different types of samples, bacterial, plant, and human samples. And this was largely enabled through the new SQL chemistry, which we call version 1.2.1, which we have announced this week, which in addition to giving more robust and reliable performance, also significantly improves on the amount of library that is needed to run per SMART cell. So right now with the new chemistry, the on-plate concentrations are between 10 and 30 picomolar, so on par with what you're used to from the PacBio RS2. And that is about a 50-fold reduction in the input requirement compared to the earliest SQL chemistry, making a lot of projects feasible. We realized that this high input requirement was in some cases prohibitive for really running projects and also cost prohibitive. And with this reduction and the removal of that bottleneck, we certainly hope that this will now allow a lot of projects to go forward. So this chemistry is available for ordering as of today, and we'll start shipping that next Tuesday. I've highlighted here in bold some of the runs, the bacterial runs and human runs, just to highlight that we don't only get this kind of performance in the lab, in our site, but this is runs from external SQL users on their libraries, on their runs, and we've seen very consistent performance there. So lastly, I would like to spend a few moments on talking about how this PacBio data and these capabilities compare to other approaches. We get a lot of questions and a lot of interest. How does PacBio capabilities compare with Illumina, and then also some of the newer linked read approaches, molecular and more recently 10x Genomics. And so there's a number of publicly available data sets and presentations and results available. So I thought it would be of interest to share these kinds of comparisons with you and some of the analysis. So with regard to alignment-based calling, NA12878 is available with regard to structure variation call sets of the different technologies I already talked about, the high coverage assembly from Ali Bashir at Mount Sinai, and then the 10-fold PacBio SQL coverage. And this compares very well with the Illumina-based structure variation calls, and then also the calls that were presented at the Genome in a Bottle meeting a few weeks ago by the 10x Genomics Long Ranger structure variation caller. And you can see that in particular areas of structure variants, for example, insertions, which are much more difficult to call with short-read technologies or the short-read adapted synthetic long-read technologies, that there is a much higher sensitivity. Similarly, with regard to de novo-based assembly structure variation calling, there's also now a number of PacBio-based assemblies available, and we can compare that to assemblies that have been made available by the other technologies. In particular, this NA19240, that's the proband of a trio of Yoruban descent. Both a PacBio assembly and a 10x Genomics assembly is publicly available. And you can see here with the assembly stats that there's a much higher contiguity with regard to PacBio compared to the synthetic long-read approaches. But really what I want to focus here on is the structure variation calls that you can make from these types of assemblies. So here we're directly comparing these two assemblies with regards to deletions, insertions, and then complex structure variants. So you can see that the 10x Genomics technology calls a lot more deletions compared to PacBio, and then whereas PacBio calls a lot more insertions and complex variations. And so we want to drill a little bit down on that and investigate that a little bit further. So the reason for the higher number. So we looked at the 10x Genomics specific deletion calls, and there's about 5,600 of them, and selected randomly 10 instances of these that are in the 10x Genomics assembly, but absent in the PacBio assembly, and down here are the coordinates. One example is shown here on chromosome 1. The 10x Genomics assembly called a 176 base pair deletion. So this is shown here as 176 bases as a reference, HG38. And 0 bases in the 10x assembly. And then when you look at the PacBio reads for that same location, you see that there is no evidence for those missing bases, and they are clearly represented in the PacBio assembly and the PacBio reads that underlie the assembly. You can then ask, well, what is the reason for that deletion call? Well, this particular region is very GC rich. You can see the sequence here, about 300 bases that are 80% GC rich. And it's, of course, well known that the short-read technologies have a GC bias and struggle in GC-rich regions and AT-rich regions, and don't provide high-quality data, or in some cases no data at all, which could be the result for this erroneous deletion call. Looking at all the other sites, there's an emerging theme of, or concurrent theme of this type of phenomenon where you have missing bases that are clearly supported by the PacBio reads. In 8 of the 10 cases, in the 2 others, the deletion call was next to a complex structure variant, which likely confounded the deletion call. So then we can conversely ask, well, what about the calls that are specific to the PacBio assembly but are not present in the 10x Genomics assembly? And again, we randomly picked 10 of those cases. Here's one example where there's a clear 58 base pair heterozygous deletion called in the PacBio, from the PacBio assembly. And this happens to fall into an assembly gap. These are the 10x Genomics scaffolds, and below that are the 10x Genomics contigs. So you can see that this deletion falls square into an assembly gap from the 10x Genomics assembly. You can again ask, well, what is the likely reason for that? Well, this particular region is low complexity. So you can see here that most of the bases are A, C's, and T's. There's only 9 G residues in the entirety of this 300 base pair stretch. And again, the short-read technologies, the underlying Illumina technology that 10x Genomics uses, has difficulties spanning those regions and correctly resolving those regions. And of course, these are really important regions in the genome for short tandem repeats, variable number tandem repeats, repeat expansions that are at the heart of many neurological diseases. And I think our third speaker is going to talk a little bit about that, and Alan Rosens was talking about that. So these are quite important regions in the genome. So again, looking at all the 10 sites, a recurring theme, 9 out of the 10 randomly chosen PacBio-specific deletion calls fall into assembly gaps from the 10x Genomics assembly, and 1 was a heterozygous deletion that was missing within a 10x Genomics contig. So we find that between 70 and 80% of the PacBio-specific structure variant calls intersect with gaps in the 10x Genomics assembly, and there's about 58,000 of those in the total assembly, which highlights the difference and the importance to differentiate the concept of a contig, which is a gapless sequence without any ends interspersing and any gaps in the assembly, which is facilitated by PacBio, versus scaffolds, which is the concatenation of contigs and can have missing information. So I want to close by highlighting that we're very pleased to see the publication on Monday published in Nature Methods by Jason Chin, our Director of Bioinformatics, on the Falcon Unzip, on Falcon and Falcon Unzip, which provides, in our view, the highest quality, contiguity, and comprehensive views of human genome in the assembly context for phase-deployed genome assemblies. So with that, I'd like to turn the podium to Dr. Ewan Ashley, who I think doesn't need an introduction. He's very well known at Stanford University, and he will tell us about towards precision medicine.
Thank you. Well, thanks, Jonas, and thanks to all of you for coming today at lunchtime and making it all the way to the hotel ballroom in which we find ourselves. I'm going to talk about precision medicine. And first of all, here's my disclosures. I should mention, first of all, that I don't receive any money or consulting fees or anything from PacBio. I'm here because I think the technology is very cool and can answer a number of different challenges that I'll lay out at the beginning of the talk. And so I'm happy to be able to tell you. These days I've been we've been saying for many years, genomic medicine's coming, but really, I mean, you're at this conference, so you know this already. It's here. It's every day. We're sending exomes and genomes on a daily basis on our patients. And that was not the case several years ago, and it's worth, I think, pausing. One day we won't even need to pause because it'll just be so well accepted, but it's worth pausing to think it was not very many years ago that this was extraordinarily expensive and beyond what we could consider, and it's now a daily event that we are solving cases at a rate of around 30% for those with rare and undiagnosed disease. That just wasn't something that was happening a few years ago. But while we celebrate that, we also think about what's next, and certainly many of my talks in the last year have featured this picture of our president pointing at our green statue of the DNA and saying that he's interested in the country that eliminated polio, one of the countries that mapped the human genome to lead a new era of medicine, one that delivers the right treatment at the right time. And that's the essence of precision medicine, a phrase that most of us had not really used much until the president used it, and then we immediately went to Google to work out what he meant and started changing all our slide decks to have the word precision instead of the word personalized in it. That's certainly what I was doing. I actually prefer this quote, which is just from last week. We were lucky enough to be part of this Frontiers conference that was in Pittsburgh last week, and the president was on stage with Atul Gawande, you can see there, and he said at the beginning, you know, I'm a science geek and I make no apologies for it. And I think it was pretty clear, and if you look at the current edition of Wired magazine, which he guest edited, and he really was involved in doing that he's very interested in science and technology and pushing this forward, and he often reaches for the idea of precision medicine when people ask him what's exciting and new and innovative is coming and is coming. But precision medicine also needs to be accurate medicine, and you know that these two words have distinct meanings. But in this context, I think that we do need to think about accuracy, and it's certainly been an area that I've spent a lot of time thinking about. While we have the excitement of solving maybe 30% of cases, you know, our hearts and minds are with the 70% of people who don't have their cases solved. And we have to really test ourselves to think carefully about where the answers for those people are going to be. And this is a figure from a paper, and you're welcome to look up the paper, where we laid out a number of different areas in which I felt that the technology that has done so well in discovery-based genetics over the last few years needs to bring itself up further to be truly clinical grade. And I'm not going to go through every one of these, but just to give a few examples over the next few slides. The first one, you've seen some examples of this already. This is a capture chip actually from the former, I guess now former version of EXAC. This is KCNH2, which is a long QT gene, so a disease that can cause and be associated with sudden death. So an important gene to get right. And you can see the coverage here is very variable. And that exon towards the end that has almost no coverage, well, that would be fine if it wasn't important. But if you look on ClinVar, there are 19 potentially pathogenic variants in that. So if you can't see it, you can't call it. And we need to start asking whether we can see it or not. And so that's one of the themes. The genome is complex, and we kind of forget this because we have these fairly standard tools now that take us through, and we get variant lists. And certainly those of us who are at the end of trying to solve these issues are often looking at at variant call lists and thinking less about what has happened before that Think about the repeat masking. Think about what happens to those short reads that actually would map to multiple different places, and what decisions have been made either at the command line or by somebody in our bioinformatics team as to what happens with those. And we need, I think, to continue to ask those questions. Even in the absence of that 100 base pair reads, any normal genome would originate 5% of non-unique 100 base pair reads. So even in the very best, completely error-free situation, then 5% of the reads are going to be non-unique. Also, repeat tracks cause disease. And here is a nice summary from this Annual Reviews in Biochemistry article, showing that if you look in the red area for all these diseases, the repeat length at which most diseases called is up in the 200 to 2,000 base pair range. And 100 base pair read is going to be quite challenged to really sort that out for the vast majority of patients whose disease is caused by repeats in that kind of length. Also a reminder that we're very good at single nucleotide variation because of 1,000 genomes and the focus on that through the GWAS era. We actually have great algorithms and always have for calling those from sequence data, but we haven't spent nearly the same time thinking about calling small indels, large indels, or structural variants, and we've bemoaned that. But as Jonas pointed out earlier, a large amount of the variation in the genome that we're interested in that is disease-causing clearly is caused by disruptions of the open reading frame that we're not good at calling. This is one example from a paper we published a couple of years ago looking at these are actually calls concordant or discordant between Illumina and complete genomics. But just to give an example, when you think about insertions and deletions, it's down at 50%, it's even 30% through the whole genome, as opposed to the 99% that we expect when you have single nucleotide variation. So we just need to pay more attention, and that can even be algorithmic. There's some nice algorithmic solutions to this. But we have to, I think, be creative about how we get to that. So what are some of the potential solutions? Well, the first thing is if you've got a hole, potentially you could just fill it in. And so that is certainly something that companies like Personalis, but others, the Baylor lab has done this, and groups of others have said we can just capture more in that region and that can help us. So thinking about augmented exomes would be an important first step. Here's a clinical quality metric that we've defined. It's really the way the community is going, which is to say that if we really need to know how many base pairs are not covered at 20-fold at a quality of Q20 and preferably uniquely mapped. Because that's the non-inferiority with Sanger sequencing. That's where the clinical world is right now. You don't sign out an exon unless it's covered, unless you call every base pair. And we moved into this world, which is very exciting, of high throughput testing, but we moved it into the clinical world without maintaining that clinical quality metric. Which is to say that if there are many, many base pairs in an important gene that you can't call, we should know that. And this kind of graph is one of the ways of looking at that. It's a little small for you to see, but the x-axis on each of these graphs is the bases that are not covered. A few different platforms are shown. And then the genes that are listed on the horizontal, on the y-axis, are the 56 ACMG genes. So if we really have true quality clinical grade for each of these genes that the ACMG has decided are the most important for clinical medicine, then we should have a straight line down to zero. We should cover every base pair of each of those with high confidence. But as you can see, it's nothing like that. The off-the-shelf exome is truly terrible from that perspective. The whole genome, short read, high-seq, and X10. Actually, the X10 is a little worse than the high-seq on this particular metric. And for these genes, the filled-in genome does better. Thinking about how we do that on a genome exome basis, you can see if you just look at the graph on the right, if you want a whole genome short read this is to equal the coverage of these augmented exomes, you're going to have to be up around the 50x mark, not the standard 30x mark that is sort of the clinical short read genome. And there's a bioRxiv paper on that if you're interested in more details. What about those low complexity regions, though? These are going to be very challenging to cover. I mean, there are approaches with extra fill-in and algorithmic approaches to some of these, but these low complexity regions aren't going to be solved with that approach. And this is an example from the long reads from PacBio, of course, in the RPGR gene, the retinitis pigmentosa GTPase, showing that big hole there with the Personalis graph shown in the middle, but also showing very clearly that the long reads from PacBio are able to cover that region and allow calls. That region is particularly avid also for pathogenic calls. And you may have seen this before from the Hypertrophic Cardiomyopathy paper from Evan Eichler, but just a sense, and Jonas gave you a sense of this earlier, of just how many novel deletions and insertions that you find when you start to use this technology. And this is the sort of thing that keeps me up at night. I think about, I'm in a cold sweat thinking about what we're missing. And there's a lot that we're missing currently. So, well, what about clinical application? I wanted to, I'm a cardiologist and a geneticist at Stanford. I help run the clinical genome service there. And we're obviously very interested in technology just because we want to do a better job for our patients. And so I wanted to tell you about one patient of mine, a 22-year-old male, and he had a syndrome that wasn't incredibly well documented and not incredibly clear exactly what it was. He had hyperpigmentation, he had microadenomas of his adrenal and pituitary, so totally Leydig cells of the tumor of the testis. But the clear and most important finding from his clinical presentation is shown in the videos on the right, which is cardiac myxomas. And to give you a sense of this, if I can get my pointer here, it's not going to be very clear, but if you look over here, there is a small round something there in the middle of this patient's heart, which is wobbling in the outflow tract. So the blood comes into the heart through here, goes into the main chamber, and then leaves the heart through here. So in the outflow tract of this patient's heart is a large tumor. We're not normally used to thinking of the heart as having tumors because the cells are terminally differentiated. But this is the major problem with the patients who present in this way. If you also look at this thing that appears here at every beat, that's a much larger tumor that's also in the right atrium, which is the upper chambers of the heart. So very early on, from the age of 4, this particular patient had open heart surgery to remove these myxomas, but the problem is that they come back. And the challenge now, as he's being considered for heart transplant, is what does he actually have? Because if you're going to put someone on the transplant list, it's going to be very important to know what the condition is. He certainly has a clinical picture suggested of Carney complex, which is a multiple neoplasia syndrome. But clinical testing was negative for the one gene that is known to cause that. So with help from our collaborators at PacBio, and this analysis I'll show you the next few slides was carried out by them. We look forward to playing with the data very soon, but as you'll see from the date, it was carried out just in the last couple of weeks. So this is also the prior chemistry. Jonas talked about the newest chemistry. This is one prior, so the input DNA is higher for that reason. And this is 10 SMRT cells again. The yield, as you'll remember from just a few minutes ago, is now higher per flow cell, so this would be fewer SMRT cells required. And here's the read length N50 at the 9,000 range, and you saw that also going up with the new chemistry. Here's the variant size graph, or I'll let me word quickly on the tools. Of course, it's important, but these are clearly coming now and being developed. This tool by Mike Schatz and Adam English on PB Honey, these specific tools for variant calling, this one using a modified Smith-Waterman, but basically looking and acknowledging the error profile of these long reads and then optimizing the calls. You can't just plug and play the BWA GATK approach and the IGV viewer even that most of us have used. And you can see obviously the variant size shown here, the allele repeats shown here. And here's the sorts of findings that there were. If we think about these structural variant calls being optimized above 50 base pairs, we certainly could have a conversation about the sort of 3 to 50 base pair range. At the moment, we of course lack an EXAC or a NOMAD, I guess, as we're now going to start calling that resource. At the moment, we have the 12878 healthy controlled genome, and that can take us from the initial call set down quite considerably, but notably it doesn't take us down nearly as far as we'd like, and I think the community is in urgent need of an EXAC-like control population for structural variation, and that's something I think we need to think about and talk about. Now we can detect these, we need to know what's common and what's not. But as you can see, even with this filtering that we have today, we were able to get down to 6 structural variants that were linked in a gene that were linked to a disease, and this is them shown here. And importantly, if we lay those out, then one of these in particular is actually the gene associated with Carney complex. So we think of this as a very strong candidate for this condition, and shown here with the IGV viewer is the coverage. You'll note that in that area there's basically 4 reads, and while this is a kind of 8 or 9-fold coverage genome overall, because we're looking for structural variants and because the error profile is on a more of a per base level in small insertions and deletions and single nucleotide variants, you actually have very high specificity for these much larger structural variants such as this shown here. And we have actually confirmed in this particular patient through an orthogonal technology that there is a 2184 base pair deletion in that region that we believe taking out exon 1 of the PRKR1 gene, 1A gene, is actually responsible for this condition. So I think that showing the technology and showing the numbers is one thing, but it really comes alive for us at the level of finding something for a patient in whom we are still making clinical decisions. This is not a clinical test shown here, it's a demonstration. But we're very interested at Stanford in particular of looking at what the new technology is able to bring, and I know you'll hear some other examples of moving the technology towards the clinic going forward. So just in the last few minutes, I'd like to tell you just a little story of some work we've been doing in the lab. This time not on the genomic DNA side, but on the RNA side, but also very focused on a translational target, which is hypertrophic cardiomyopathy. This is a Mendelian disease of the heart muscle. It's mutations in the molecular motor of the heart. Myosin heavy chain, the MY7 gene, is a common culprit. And the heart changes in the way that you see here. The normal heart, if you look shown on the right, shown on the left, but the right-hand panel here is here with a normal-sized chamber and normal fibers. Here you see the fibrosis of hypertrophic cardiomyopathy and the thick wall here. This is one of the most common conditions associated with sudden death in young people and in athletes. And so you may have heard of the disease through high-profile deaths of young athletes. And here's some moving pictures from my patients in our center at Stanford. And you can see from the MRIs in the top panel and the echocardiograms in the bottom panel, the sorts of changes, a thick wall shown here, a thick wall here at the apex of the heart, an obstruction to blood flow shown here, and here and even an aneurysmal segment of the heart as a result of the thickening of the mid-cavity chamber shown here. So it's a significant and severe disease that currently has only really palliative therapy. And believe it or not, this is true, right? The way we treat this right now is to either do open heart surgery and chop out a piece of the heart called cardiac myectomy, or we quite literally give a so-called therapeutic heart attack. We go in with a catheter and we inject alcohol directly into the bloodstream. Some of you may have been doing something similar at the parties last night, but this is actually in the coronary arteries, and the point is killing a part of the heart muscle, as you can see here shown in blue. And so, when your therapies are chopping out part of the heart or causing a therapeutic heart attack, you know that we have some work to do in getting better. And so we've been focused on that and thinking about the genetic understanding of the disease leading to a genetically guided therapy. But of course, we know that variation is rare in general, and the site frequency spectrum for this is very much like other Mendelian diseases where we see recurrent variants rarely. And so do we really start to think of a day where we're making a therapy directly for every given individual? Or can we think about using knowledge of haplotypes, personal haplotypes, to target common variation? And so if we model that, for example, for this disease, let's take MY7, which is the myosin heavy chain, and if you wanted to go after, let's say, 47 HCM patients here with these unique disease-causing variants, you'd need 34 unique therapies. But if you want to target the common SNPs that are on the same haplotype, then you can get most of the gene and therefore get most of the patients covered with just 5. And so if we include haplotyping as part of our therapeutic development, this is work shown here from Alex Danis in my lab using IsoSeq. You can use the long-read approach to basically do personalized haplotyping. These are some examples of actual patient samples. And phase SNPs, of course, will benefit from the new IGV viewer here as well for doing this going forward. But basically proof of principle that you can provide personal haplotyping and then connect that directly to a precision therapy. And this is some of the precision therapy, in this case using shRNA to silence the mutant allele. Shown here, the SAR403Q is one of the most common mutations that cause hypertrophic cardiomyopathy. And we can show in iPS cells that we can silence both the phenotype and, of course, the gene expression of the mutant. And we can do that by targeting both the rare variant, as shown here, and this is particularly successful in this case, a little less successful in targeting the common variant, but still 50% reduction in the expression of the mutant allele, we know, or we have good reason to believe, translates to a significant therapeutic benefit. So I think in future we should be able to tie together this better diagnostic technology and personal haplotyping with the idea of precision targeting of the actual diseased haplotype of the individual. And I think that's an exciting new direction for us to go. Well, in conclusion, I think I've tried to convince you anyway that we need to spend some time thinking about the accuracy of genomic medicine. I think we have a lot to celebrate in how far we've come, that genomic medicine is here and something we're doing every day. But really to push us to the next level and to think about where we go next, we have to go back to acknowledging the complexity of the genome and look at the limitations of some of the short read approaches and think about creatively wherever we need to look for technology that can improve things. We can improve coverage of key genes. That's a straightforward way to start. We may want to add some whole genome coverage along with these, with some of the exome approaches so that we can call structural variants better. And long reads, I think, clearly have a role to play in that. And ultimately, complexity in areas of the genome is clearly associated with disease. And so I think embracing that complexity is something that we have to do to move us to the next level. So just wanted to thank the people who pay for us to do this and the people who do the actual work shown here in the picture. And thank you for coming and spending your lunchtime, spending a little bit of time with us today. Thank you.
Thank you very much, Ewan. In the interest of time, we'll keep going, and if we have time, there for questions and answers at the end of the workshop. The speakers are also available after the workshop if you have specific questions. So our next speaker is Dr. Melissa Laird-Smith from Mount Sinai, and she will tell us about, I think, quite a large variety of different applications of the technology. Melissa.
So I want to first take the opportunity to thank all of you for sharing your lunchtime with me, and also PacBio for inviting me to speak today. I recently joined the Icahn School of Medicine at Mount Sinai about 4 months ago, and quickly dove into the technology development group there as their assistant director. So the Mount Sinai Health system is enormous. It's one of the largest healthcare systems in New York City. As such, the people doing basic research there have the opportunity to interface with an incredible number of clinicians, and the population of New York City alone provides an incredibly complex group of people from which to do a lot of very interesting clinical research and associations and healthcare-related research. So in that circumstance, we run a technology development group at the Institute of Genomics and Multiscale Biology there. And as the demands of the health system require, we have quite a large toolbox to play with as far as genomics tools to look at associations with disease and basic biomedical research. So currently, this is the list of the instruments we have in the technology group, and most importantly for today, and something we're very excited about, is that we received the first 2 PacBio sequels earlier this year, and have been working with PacBio to optimize the platform in the last several months. So using the variety of different tools that we have, we really are focusing on long-read sequencing as a translational research tool. Again, we have multiple technologies that feed into that pathway. And we're using those long reads to assess complex structural variation and targeted sequencing for comprehensive diagnostic development. We're also doing rapid validation of low-frequency somatic variants in heterogeneous tissue and cancer samples. And something near and dear to my own heart that I won't have an opportunity to speak on today are rapid and complete infectious disease surveillance, although I will touch a bit on immune repertoire profiling. So I'm going to jump right in. I'm going to tell a few very short vignettes about a diverse association of projects that we've actually transferred onto the SQL system, so I can show you some SQL data, as well as how we're using targeted amplicon sequencing to address variants in disease using long-read sequencing. So we know from Jonas and Ewan that there are a number of loci with known disease associations that require long reads. Many of these are due to pseudogene complexities with assembly and association variants. There's several repeat expansion disorders that Jonas and Alan Roses touched on in that wonderful video, and then several also of extremely polymorphic loci where the phasing of variants across is very important for discriminating the association with disease. So this is sort of an overview of several of the projects we have in the lab in cancer, immunology, inherited disease, personalized medicine, precision medicine, and pathogen surveillance. Those highlighted in pink are the ones that I'll touch on today, again, in sort of short vignette format. So the first I want to touch on is the CYP2D6 project, recently published by Stuart Scott, who I think is out in the audience, a great collaborator in the clinical genomics group. CYP2D6 is a gene that metabolizes about a quarter of all medications, and these include antidepressants, antipsychotics, opiates, adrenoreceptor blockers, et cetera. There are over 100 alleles identified, many of which include copy number variants. So obviously resolving these variants is very important to understand the mechanism behind CYP2D6's relation with drug interactions. And so the benefit of CYP2D6 is that the long reads will encompass the entire allele, allow us to identify novel alleles, and further resolve the variation within that locus. So just to show you a little bit of data, actually this is run on the sequel last week. So this is sequel coverage across the CYP2D6 amplicon that we generate. It's about a 6 kb amplicon. It's very hard to see the axes here, but that axis is, on the y-axis is coverage, and you're looking at between 400,000 and 500,000 X coverage across this amplicon. So obviously overkill, but exemplary for this audience. So the sequel statistics that we're getting now, we're getting almost 400,000 mapped polymerase reads to this amplicon. The polymerase read N50 is over 12 kb. And obviously, with such high coverage, we can now start talking about a high-level multiplexing. So with the over 384 barcodes available from PacBio currently, we could multiplex CYP2D6 per SMRT cell at that level, with still a lot more bandwidth to then pool different genes on that same chip. So again, looking at these data, this was taken actually from the paper that Stuart published earlier this year, I guess last year, and the resolution of the SMRT sequencing of CYP2D6 not only confirmed haplotypes that were seen with other technologies, but also were able to resolve ambiguous alleles and even identify novel variants within these samples that were screened. So I'm going to sort of quickly move on to another vignette here. So this is, we're going to talk a little bit about Fabry's disease, which is a disease spectrum that's due to varied mutations in the GLA gene. So Fabry's disease itself manifests as a range of systemic symptomatic symptoms, which are often misdiagnosed in childhood because of the scattered orientation. So this includes kidney failure, cardiac arrest, also a lot of dermatological symptoms, fatigue, nonspecific pain. It actually is due to a deficiency in the alpha-galactidase, the GLA gene, which results in insufficient lipid metabolism in the body, which it causes the accumulation of lipids, which results in widespread organ damage. So the spectrum of these symptoms and severities appears to be due to multiple different pathogenic mutations scattered across the 7 exons of this gene, and the penetrance. So it's an X-linked syndrome, and so the incomplete penetrance in women in particular can lead to a range of different symptoms. So as you can see from the figure I just pulled up, these are SNPs that have been associated with different pathogenic manifestations of this disease scattered across the 7 exons. And when those are modeled onto a crystal structure of the GLA gene itself, you can see they're scattered throughout the protein. Meaning that really being able to phase these mutations across the gene itself would be very helpful in trying to identify novel variations within this gene. So again, we use a PCR amplicon sequencing-based approach. Again, this is a 6.2 kb amplicon that we're running on the sequel system. Just in running this, we identify using the PacBio mapping and variant calling algorithm a panel of variants here that are so many as to be completely invisible to you as the audience. Obviously, they would require validation, but you can see that there's quite a lot of information that just comes out of sequencing data directly. The sequel data that we've obtained recently, again, we're at 300,000 map polymerase reads with about a 9 kb polymerase read N50. And again, as another 6 kb amplicon, this could be something very interesting to multiplex along with other loci for these individuals. All right, so I'm going to jump again to glioblastoma. So glioblastoma clonal evolution is shaped by selection pressures and mutagenesis during therapy. That therapy, standard therapy, includes radiotherapy, which can induce DNA double-stranded breaks. So in collaboration with Raymond Young, we've worked on an inverted PCR technique that allows us to genomically localize these double-stranded breaks with the idea of helping to predict the acquisition of breaks that may underlie the emergence of resistance. So this PCR is schematized to the right, and basically the primers lie in such a way that when these double-stranded breaks are made and repaired, we can identify the different breakpoints using a long-read sequencing approach that spans these breakpoints. So here's a little bit of data that shows this. So this is the alignment of that exon. The breakpoints are highlighted in these arrows, and when you run control versus irradiated samples, you can see the ability to call de novo breakpoints just using the sequencing data. So the control samples are below. You see a few breakpoints here at position 730 and position 1150. But when you look at the irradiated sample, you see a much noisier graph because there's far more breakpoints at different points across the exon. So I'm going well within time, going quite rapidly. I apologize for that. The next thing I want to talk about a little bit is validating somatic variants in sort of personalized cancer therapy treatments that we're developing in our group. So this is a complex pipeline. This is a paper published earlier this year by Andrew Uzalov, a close collaborator of our team. And what Andrew's been doing is using a series of different assays to screen cancer patients and identify variants that may be associated with the reaction and receptiveness to therapy. That includes whole exome sequencing or targeted panels on the ion torrent. And then we need to then validate these variants as actually relevant to the pathway and in our workflow here. And so we use PacBio to do that validation of any of the low coverage or ambiguous variants that were called with the other technologies. So an orthogonal validation of these variants for clinical research purposes. So what I'm showing you here is one of these variants. This is a key driver mutation in medullary thyroid carcinoma. We ran this data both on the PacBio RS2 system as well as the new sequel system, and you can see the comparison of the data here. And then we looked at the ability of the two systems to call the different variants in our samples, and you see that the variant calling for the RS2 and sequel systems are very similar in the relative percentage of those variants within the population. Again, we recently ran these on the sequel. This is a 250 base pair amplicon, so a very small amplicon. We're getting almost 400,000 CCS2 reads. So these are the circular consensus sequencing reads where we've gone over the molecule multiple times to generate an incredibly highly accurate sequence. So these have an average of over 20 passes per molecule. These reads are over 20 kb polymerase N50, and we generated 6 gigabases of data in this single SMRT cell, which was by far enough to call these variants. So my last vignette is looking at immune profiling using SMRT sequencing. This is very near and dear to my heart as a molecular virologist and immunologist. And it's something that I'm very excited that we're sort of getting up and running in the lab. This is very early pilot data that I'm showing you here. So we would like to use the long-read sequencing and that circular consensus sequencing approach to generate full-length single-molecule VDJ data, so spanning the V, D, and J segments of B-cell and T-cell receptors. Immune profiling in particular can identify particular T and B-cell receptors that are responsible for the protection and in cancer immunotherapy or critical infectious diseases. And T and B cell populations are extremely variable in all of us, insofar as they have to detect any sort of threat that you receive from anything non-self, any antigens or non-specific pathogens. So the way the body does this is a very complex germline-based rearrangement system where your body recombines a single V, D, and J segment from a selection of them that are encoded in each of our genomes into a rearranged VDJ segment, which encodes the antigen binding portion of a T or B cell receptor. And there are multiple mechanisms of variation here that make this population incredibly variable. So we have 10 to the 9th different T cell receptors and around 10 to the 12th theoretical B cell receptors circulating in all of us at all times. So the random mixing of V, D, and J segments, the addition of non-templated nucleotides at the V, D, and J junctions, the specific associations of different VDJs for heavy chains and V and Js for light chains, and then additionally in B cells you have an extra layer of hypermutation upon maturation. So we believe that utilizing SMRT sequencing will generate single molecule passes that are contiguous through this incredibly complex environment to generate highly accurate full-length T and B cell receptor sequences that require no reconstruction or imputation of overlapping reads. So in this case, I'm going to show you an example of T cell receptor sequencing. So we use the recently released CloneTech Smarter Human T cell receptor alpha beta profiling kit, which generates these full-length amplicons. They're about 700 to 800 base pairs. We piloted this assay using the Jerkit T cell line that's widely available from ATCC, and we ran these data on a sequel. And what you can see is that we're getting, again, a huge number of reads from the sequel, generating about 7 gigabases of data in a single chip. Now, if you use the default parameters of that circular consensus sequencing algorithm and filter at sort of 90% accuracy, we get about 400,000 reads. But as these are a very highly variable population, you want the most accurate reads possible. And so if you increase the read accuracy filter on the CCS algorithm to a Q30 or a 99.9% accurate threshold, you still get out about 236,000 reads that can then be used in the mixer pipeline to identify those receptor sequences. So this is very novel. This is very new data. It's sort of hot off the presses 2 days ago, but we're very excited about the potential to apply this also to B cell receptors and do a lot of immune profiling using the PacBio platform. Sorry about that. So I would like to stop and thank all of our collaborators, Bobby Sebra, who's the Director of Technology Development, Eric Schott, who's the head of the Institute of Biology and Multiscale Genomics at Mount Sinai, all of our wonderful collaborators, and then my new and awesome technology development team at Sinai who makes this all possible. And thank you all for your attention. I'll be happy to take questions after the end of the presentation. Thank you very much.
Thank you very much, Melissa. And our last speaker is Dr. Mike Lutz from Duke University, who has agreed to speak on Alan Rose's behalf. We're very grateful for that, and we look forward to your talk.
Thank you. I'd like to start by thanking the organizers for the opportunity to present this work today, and to thank you for attending this workshop. So in the video, you heard that one of the topics that Alan was particularly excited about, and I was fortunate actually to work with Alan, Dr. Alan Roses, for many years, both at Duke and before that at GSK. But one of the topics that he was very enthusiastic about recently is the identification of highly polymorphic informative structural variants associated with neurodegenerative diseases, including Alzheimer's disease, Lewy body dementia, and ALS. Today what I'd like to do is describe a strategy and an illustrative example that shows how this can be done in practice, really get into the details of how this can be done. The interest in these variants is really twofold. First, to use them as biomarkers, genetic biomarkers for disease and pharmacogenetics. And second, as Alan pointed out in the video, to bring you closer to mechanism, particularly where the mechanism involves regulation, gene expression, and splicing. You'll see, as I describe the strategy, that it involves a combination of bioinformatics, computational approaches, sequencing, and biological work to do the validation of the variants. And I do want to focus on those aspects of the strategy that are particularly well suited for long-range sequencing, because that's really a key aspect of this. I will point out that the software that we use to facilitate the process, we've published this recently with our collaborator, Bob Saul from Polymorphic DNA Technologies. The reference is there. It's public domain software, so it's freely accessible and usable. I won't have time to cover phylogenetic analysis for genetic association studies. Alan mentioned this in the video clip. However, I will point out two things. One is there's a publication that gets into some of the details that I list there. And second, that this is also a really great application for the SMART sequencing technologies because of the length of the sequence that's involved. So by way of background, and you've heard from many speakers in this ASHG meeting, genome-wide association studies have identified SNPs and regions of the genome associated with neurodegenerative diseases. However, translation to causal variants and clinically relevant biomarkers hasn't followed. The hypothesis that really underlies this work is that changes in expression levels of disease genes is an important mechanism that underlies the disease etiology, and that specific causal SSVs modulate the expression of these disease genes. This is very often, it involves the SSVs acting as enhancer elements. And if you went to the opening plenary, you would have seen a nice example of enhancer elements in cardiovascular disease. So I think there's a lot to look at in terms of how approaches that are used in specific clinical areas translate to other areas between, say, cardiology and neurodegenerative diseases. We have a very broad definition of structural variants, and a publication in Nature Reviewed Genetics some years ago, I think, did a great job of describing non-SNP variation and its impact in various human diseases. This was a paper by Fraser et al. We actually take that a step further and focus specifically on short sequence repeats, which are variable lengths of the same nucleotide that are highly polymorphic, and short tandem repeats. And short tandem repeats are well precedented in neurological diseases. For example, the hexamer repeat C9orf72, which is implicated in ALS and frontotemporal dementia. I think it's very helpful to consider this framework shown on this slide for the interpretation of genetic variation. So in terms of allele frequency, how often do the specific alleles occur in different populations and in different cohorts, either individuals with the disease, different ethnicities, control individuals, and then their effect size. And the GWAS studies, and primarily the Alzheimer's disease GWAS studies, really occupy this quadrant at the lower right-hand quarter, where you have common variation but small effect sizes. So effect sizes is measured by odds ratios of about 1.2 to 1.3. Examples of common variants with large effects are very few, but the poster child there is the association between the ApoE genotype and Alzheimer's disease. What we're finding with the structural variants that I'm talking about today, and this is really covering a few different areas, including ALS, Alzheimer's, and the example I'm going to talk about, Alzheimer's disease with Lewy bodies, is really a diagonal, whoops, if I can go back, is a diagonal that stretches from the lower right corner through the middle. So we do see variants that have the fairly common frequency and low effect size, just like GWAS, but we also work our way up to the middle, less common with moderate effects. So effect sizes, again, of about 3 to 5 in terms of an odds ratio. So this slide describes the strategy, and as I mentioned earlier, there are really 3 pieces to it. First, the bioinformatics, which is involved in identification and prioritization of potential variants. The smart sequencing, targeted sequencing is the second step, and this is where this work is done to identify whether or not there are associations between either individual SSVs or combinations of SSVs with the phenotype of interest. And finally, validation of mechanism to really show causality, animal models, CRISPR-Cas9 gene editing, gene expression experiments are all done to actually validate that a variant is indeed causal. Don't have a lot of time to talk about the software. The software is probably the biggest genome-wide aspect of this, which is using a lot of different public domain data to map to specific tracks that we then use depending on the specific study that you're looking at. So the GWAS signal itself is a very important aspect very often to the bioinformatics. We also look very carefully at linkage disequilibrium to try to disentangle the relationship between specific variants that are in LD. For studies that are focused on gene regulation, we use data from ENCODE, and for other types of tracks include transcription factor binding sites and the context of the repeats. So here's the example, and this example is collaborative work with Dr. Oneet Sheba-Falek at Duke and her laboratory. And it's an example that really involves the heterogeneity of disease, specifically the Lewy body variant form of AD. So in this case, you can think about control individuals as having Alzheimer's disease and your cases as having the Lewy body version of Alzheimer's disease, or another way of putting that, your controls are absent of Lewy body version of Alzheimer's disease. And it's important to keep in mind that to do this, your samples have to have very clear histopathology, neuropathology, and with the Duke Brain Bank, we had that available for this study. So where to start? Oneat had published some work earlier on a complex repeat, REP1, in the alpha-synuclein gene, or SNCA, that was associated with Parkinson's disease and also with changes in gene expression. So the work from Parkinson's disease, we were considering how could that translate potentially to Alzheimer's disease, and specifically the Lewy body version of it. So this chart here shows you some of the kinds of things that we thought about in terms of choosing regions to search. You could see a region of high linkage disequilibrium on the left-hand side of the screen. That was potentially interesting. And then we also looked for regulatory signals, since we were particularly interested in repeats and structural variants that might impact gene regulation. The bioinformatics step is fairly fast, which allows you to run a lot of different simulations. Effectively, the data sources that I showed you earlier are weighted and used to derive an impact score, an empirical impact score. The higher score would be hypothesized to be more likely to have a biological effect. And again, this covers all sorts of different structural variation, including really large repeats, as well as relatively small indels, SSRs, and short tandem repeats. The next step, and this is the step that really is best suited for the SMRT sequencing, is the association with the phenotype. So again, what we had were samples where we had a phenotype of either Lewy body version of Alzheimer's disease, or LBV, we'll call it that, versus Alzheimer's disease. And although we did not do full sequencing yet at this stage, we had prior SNP data that showed that the same location that was predicted by the search also corresponded to a very high signal in terms of differentiating these two forms of the disease, an odds ratio of 2.6. So by virtue of the fact that the location was the same between the two, we wanted to dig into that region in more detail, and that's where the sequencing came in, again going back to the strategy. So I have to fast forward over a number of different iterations between the bioinformatics, the sequencing, haplotype analysis. It turns out that there wasn't a single SSV that really differentiated these two forms of the disease, but in fact it was a haplotype, and some of the talks earlier got into some of these low complexity regions, and indeed that's what seemed to be the case here, that there were 4 particular haplotypes that seemed to be important and that we wanted to go on and then test. One thing that was interesting in the publication that covers this data I show on the bottom of the slide, there were 9 variant sites in this haplotype, which would give you 512 possible haplotypes, but we only observed 4 in this data, which is fairly rare. Usually with haplotyping, you have so many it becomes hard to understand what the associations are. But with the 4, we could look specifically to see whether or not there was an association with our phenotype of interest. We did observe a strong association between the haplotype and the LBV phenotype. You can see in this mosaic plot that haplotype 3 was entirely associated with the ADLB phenotype. Haplotype H2 greatly enriched for it, and the reverse is seen for haplotype H1 and H4. So now we get into the biology, and I should point out that haplotype is in the 5' region of intron 4 of the alpha-synuclein gene. Into the biology, the first question is the disease relevance of changes in gene expression. So we had some prior data on this, and we wanted to try to confirm it, again, looking at this disease heterogeneity. So we asked, is there a difference in individuals, samples from people who have the Lewy body version of the disease, as you see on the bar on the right, with the Alzheimer's disease controls. And indeed, the Lewy body pathology does co-occur with increased levels of the alpha-synuclein messenger RNA. The next step was looking for a haplotype association, and that's what's shown on this slide. So haplotype 3, which you saw earlier, was significantly associated with alpha-synuclein mRNA expression levels. There was relatively few homozygote carriers of haplotype 3. However, the haplotype 3 heterozygotes were far more common, and they also had a significantly higher expression of the alpha-synuclein mRNA. So this has then some potential as a biomarker for the disease. In summary, this sums up some of the major points about the motivation for the work, using the bioinformatics software to identify candidate regions, and the use of targeted sequencing. I do want to focus just very briefly on the PacBio, the value of the PacBio data to this work. So a different project, this is more specifically associated with Alzheimer's disease risk and age of onset that people develop the disease. What's been clear from some of our more recent work is that specific haplotypes of ApoE alleles and a poly-T in the TOM40 gene are associated with a number of aspects of the disease presentation, including risk, age of onset, and rate of cognitive decline. But it's been difficult in the past to analyze these relationships because you couldn't put them in phase. You couldn't put these variants in phase. And indeed, when we have been able to phase the data, we see some very interesting patterns in the data with respect to ethnicity, for example, that have helped us actually to derive where some of these ancestral haplotypes originated. But the PacBio long-range sequencing allows you to phase these haplotypes across multiple genes, and in this case, across ApoE and the poly-T that we had pulled out many years earlier as being important. So this really was a promising way to look at these specific haplotypes. Another aspect is that we have to be able to call the poly-T length extremely accurately. And this view of the data shows you that the PacBio long reads can resolve structural variants, including contractions and expansions and other tandem repeats very, very clearly and accurately. And this is not done very well with the Illumina short reads. And in fact, any attempts we had made to try to assay the 523 allele with most existing methods really didn't work and didn't give the accuracy that we needed. So finally, I just want to close with acknowledgments. Again, Alan, the driving force behind a lot of this work, Onichi Bafalek and her laboratory, who I've collaborated with, Bob Saul, who helped develop the software for that I described today. And with that, I'll thank you, and I guess I'll turn it back over to the organizers.
Thank you, Mike. I'd like to thank you all for coming. As I mentioned, the speakers will be available for questions and answers. Please stop by our booth, number 718, and we look forward to talking to all of you about your research areas and how smart sequencing can contribute. Thank you so much for coming.